I might
So then over
turn in
here,
auto head.
I'm going to do my dive status off deck.
And
Submit,
I should...
just do that real quick, and then we're right back here. I'm watching what's going on with the vehicle in relation to the ship.
Yeah, that's
Ship's
why we
going
waited.
forward. You can see that it is
Hey, Rennie,
streaming
can you
aft. Yes,
stop four-stalking me?
Roger. All good.
Thanks. And I should not be four-stalking you.
Yeah, that's all good. I'm listening anyway. Copy, clear of the transom.
So that's why we waited on deck. I was taking my time. I was waiting for that to stabilize. And I can either turn and burn, or I can lateral, or I can do nothing. So doing nothing obviously takes a lot longer, and our temperatures will be going up. So motor's getting hot, hot, hot.
Yeah, I expect that that current will be taking you.
And so I need to probably, what Rennie's hinting at is I need to do something here. And Robert will get really aggravated with me if I stay out here too long, because he's convinced I'll break the BSR.
We did that once.
Again, so I'll break the BSR again.
Yeah.
So at this point I should have full power, and I'm actively driving.
Yeah, copy that. We are making our way over to center.
So I've got full laterals on there and I'm burning, but my auto heading is wonky, so I'll double tap it, because one was full thrust forward and back, which is not helping. It looks worse on this camera than it is on this one. And I can tell you right now, Robert's going to give me grief because I didn't get the goal post fast enough. But I'm doing this a bit on purpose to do it out of the box. Right. Not too far out of the box, but yeah.
So you guys don't necessarily drive to center line, you just sort of...
We do. I'm just showing you that you can not drive the center line. The currents get me a little, so it's I'm thrusting over as fast as I can. The other thing is, you know, there's no hurry. I can take as long as I want here.
That's Atalanta going overboard there.
Yep.
So at this point, once Atalanta's going off, I should be full forward because I'm the third tag line. So once it lifts up, once it's clear of the deck, I'm pulling on it. But our forward way, our forward motion.
When you're, let me know when you're clear for mezzos.
Yeah, clear for mezzos. Okay, you gonna say it? I'm waiting for, usually, that's Hercules dive, dive, dive.
Hercules, dive, dive, dive.
That's a tradition.
Do you want me to remind you about the tether apps?
Yes, please.
Tether apps.
Tether apps.
So now I'll zero my tether.
Roger.
And they're in the water, so I'm going to do that.
Either do it for them, or I'll go to
Turn
Atalanta
those
and
on.
I'll zero the 6-8
So
reps.
we'll get Atalanta
We really
right
don't
away.
care about,
Hercules takes some time.
it's the,
But we wait for both. See, they're good, tracking good. So now I go over to ROV nav, and we let the pilot know that we've got vehicles on tracking on ROV nav.
Roger.
Clear to turn on Atalanta lights.
Turn off the stupid
Compressors.
center.
Let's wait.
Okay, Roger.
And we got them.
I need that red book, so, and I have one other. That camera's wonky.
Herc is facing the
Two
way
defects.
of the...
No, not now. We'll wait till we get through all
Yeah,
this.
you can see there's the heading of
So I've
Hercules is that red.
Turned on these
In the
cameras
compass
now,
rose.
just for fun.
Atalanta's the blue.
But what I'm really
So they're
watching here is this
opposing.
number.
We got these. We got compasses, and you have our butt cams up here. So we've got Atalanta on the left, Hercules on the right. So you can see that we are stretched out. And then we can just glance at this delta depth. That's the distance,
I don't know what's
the
going on
vertical
there.
distance between the two. So that means that Hercules is 13 meters below Atalanta in the Z direction. come over here. I'll check this
Roger.
sound speed. 1535, it's pretty hot. So I'll make sure
We get
in my
bubble
sonar
all the
dyne screen,
gauges.
which I didn't do that.
I don't know why it's freaking out.
Okay.
Stand by.
So that shouldn't change too much
I
there.
can't
See, there's not really much of a difference.
Why are you doing this?
So when we first start here, we're continuing to stream forward because we just want to make sure we're not adding anything else, any confusion by stopping the ship, right? We're going to keep streaming forward until we hit 50 meters, and we check in with the pilots if that's desired.
What I'm mostly watching right now is the aft camera. Yeah, it's pinned 100% right now.
So that's the payout right there, and then also
around
you've got
50.
Hercules depth up top. That's 53. So Herc's at 53 depth, and
What's that?
Atalanta is at.
We're
50.
right around 50.
So they're going.
You just stand by right now.
Roger.
So those are the paro depths, but then there's the payout for the winch.
Distract me.
Payout for the winch.
If you're
is what
asking a
they're
bunch of
gonna
questions
go
right
off
during
of.
the launch,
Roger.
you're gonna kill Robert. He'll bark. Trust me.
Well, yeah, that's why I'm bugging you.
Yeah, give me a minute here to keep my
Roger.
ducks in a row. Roger.
Copy that. Control van has control. Let's test it. Do a little bump there on the winch.
Yeah, Roger. Stand by.
Roger.
So
Deck confirm. We have control.
now's our time.
Now is the time.
Now we're at 50 meters.
Winch
Go
handover.
for control.
We'll just wait.
Roger.
He can hold position.
Roger that. Yeah, we can hold position here. Thank you. So that means that the Herc pilot will have some forward way to kind of maintain that tether, taut. No.
So I'm gonna.
Because if the ship's not moving forward,
You're
doing
still
that,
force
yeah,
talking
yeah.
me, right?
I am not force talking you. You're listening to SPL.
I'm gonna stop us. Thank you.
Yep.
And I'll not force talk SBL. So I'm gonna go off comms for a second.
Roger. Copy that. Bridge off radio comms.
Okay, I'm gonna jump on the comms now. So that was a launch. We're at 50 meters. The ship is stopped. The winch is stopped. I'm stopped. So we've gone from a highly dynamic situation, potentially chaotic, me taking too long to get behind the vessel, simulating massive current. That's how it's going to look if you got a big cross current, which it is going to be at Jarvis. But now we're at 50 meters, so everything's static. No winch, no boat, no ROV. And I'm a little bit deeper than I could be, maybe. When we go down, we're going to go down tail to tail and maintain 35 meter separation, almost 40. So if you look on Atalanta sonar right now, you can see a little hint of Hercules there at the 40 meter
Roger.
mark. So this tether is a little bit longer than I like, but it is what it is. The other thing we're going to watch all the way down is that tail-to-tail camera. So you should be able to see one vehicle at least in the other, and as we turn on lights and stuff.
Okay.
The reason I didn't want to turn on lights in the beginning, because daylight and we don't need them, it just would have added to the more dynamic things. And if there was a ground fault, then we would have just shoot with that, blah, blah, blah. So I don't need lights right now because I'm not looking at cameras. What I might turn on if it was night, I would have turned on the lights as soon as we went in the water. That would be a little different because the deck needs to see the vehicles and I need to see them, but I just didn't add it. It's nice to do the first launch in daylight.
HPU's Getting
hot.
warm.
Yeah, it is. And so, yeah, the motor's getting hot, and but we've got no ground fault. So at this point, we would stop. We would do an evaluate if we had any issues or anything while the deck crew's still out there crewed up. You could pull the plug, or you could come get Robert and see if Robert wants to pull the plug because it's his call.
Hey Dan, checking in on those wide field cameras. Are we good to turn those on?
They should be on.
Ooh.
Oh.
They should be on. Did I not turn them on? I thought I had them on. They maybe I didn't press that button. I'm turning them on now.
Okay, copy.
So the other thing to do is check, yeah, with nav and video, everybody's all good. We're gonna head south.
And also your ROV PTZ looks real dark. Is that expected?
No. But it might be because I don't have any lights on yet.
Okay, I saw you move it.
So I usually
There
have
it
that
is.
on the gauges at this point.
Roger.
And yeah, you can turn on all of Atalanta's lights.
Roger.
I'm gonna blast some ROV lights, make sure we got no ground faults. I leave these port and starboard off because it'll draw too many amps there.
Dan, just confirming that you know that you're still descending with Hercules.
Yeah, let's
Okay, we got the
pay out
lower.
on the winch.
We got the upper. We're good. So,
I'll pay out on the winch.
all right, we got those extra
Thanks,
cameras we
Randy.
needed.
Yeah,
Thank you.
Roger.
We good?
The wincher.
Here we go, headed south.
We good to go?
So
All right. Hi, everyone. So excited to welcome you all to the first dive of Expedition NA-163. We are diving to the
Watching the,
Makapu'u coral bed right
Separation
on the
there,
eastern side of
two
Oahu,
or three
and
boxes.
I wanted to introduce everyone
And trying to get it back in the box here. I
to
had
the control
stick
van
lock on.
today. Can we start with data logger Chris? Could you just share who you are, what you're doing, and a little bit about your job?
Sure. Yeah, hi everybody. My name is Chris. I'm a data logger. I basically record all the
breaking
information
every rule.
that we collect and take some pictures and
Make sure when that
process any
goes green, you
samples
take
we
that
get,
out.
which I don't think we're doing any for this dive.
No, no sampling today.
All right.
And stick lock too. Gotchas.
All right, science Chris? We have two Chris's on
So
this watch.
at this point, I'm back in the box. So I'm actually dragging Atalanta. My delta's back right. So now I'll go.
Is that good?
Yeah, now you're good.
Yeah, I'm a sponge taxonomist, and I'm here to help to identify all the sponges that beautiful sponges that we will see over
Full
there.
vertical down and just a little bit forward.
Great. I mean, there should be some sponges, hopefully. I'm Megan Putz. I am a guest for today's
Enough to see it
dive,
spinning.
leading this Makapu'u coral bed survey. My thesis work was on doing precious coral surveys in Hawaii, so I was asked to come and help lead this dive as one of the last people to do a survey of these types of
I'd like to
corals
be
here in Hawaii.
A little bit lower. So now the
And
two aft cameras
you all
are
heard me
there.
earlier. My name is Alice
okay,
Chow.
pilots, we're at
I
100
am
meters
the
depth and we're
Science
going to
Communication Fellow
530
on this,
meters depth.
Roger.
on this particular watch.
Can you hear me?
Oh, the port and starboard lights.
I'm Rob McCollany. I'm the ex-official watch
30
lead
is, if I turn
and
those on.
geo lead of this cruise. And I'm just here watching.
That'll go red. So over
Video, are
30
you around
amps.
to introduce yourself?
I think you were on mute.
So I'm going to turn on
Rob, I think you were
hydraulics
on
here,
mute.
and I'm going to extend the HD camera. It's usually where we run with it, all the way out.
Try again.
And I'll tilt it down a little.
Can you hear me now?
Yes. So the front row of the control room is currently very busy, working on Hercules' descent. But while we're at it, I want to talk a little bit to our scientists and what we're going to be up to today. Megan, can you introduce the coral bed to us a little bit?
You changed
Sure.
something without
So
telling me.
the precious corals
Don't do
in
that.
Hawaii, so that includes our
Don't be
pink
fiddling over
corals, the
there. Make sure I know
hemicoralliums and
what you're going to do before you
pluricoralliums.
do anything.
That
Before
is
you touch
the main
anything.
focus of the dive today that we're
What'd you do?
looking for. These precious
Don't do that.
corals are a fishery in Hawaii,
Just
and
let me
they're
know.
actively managed. Now, the fishery isn't currently active, but as part of our Hawaiian resources, we want to survey the bed and understand a little bit more about the resources we have in Hawaii. So that's why we're here today. And these precious corals you might be familiar with are used in jewelry making. So when you see pink or red coral, these are the corals that um, those jewelry pieces are made from. So they can be quite valuable, but it is also really challenging to get to the depths where these corals live at. So that's why we need a remotely operated vehicle or a human occupied vehicle like a submersible to get down to the depths and actually harvest these corals. In the past, they used dredging techniques to collect corals like this, but that's really harmful to the coral beds. So if we were to actively fish for these corals, they need to use a vehicle like ours in order to go down and collect them one by one. And so the fishery here is very closely managed and monitored, even though it's not active, to make sure that we protect our Hawaiian resources.
What are you expecting to see today?
So this bed here is actually relatively dense. So there's going to be a high number of corals per square meter. We're going to see a lot of different types of corals, mainly those pluricorallium and hemicorallium pink corals. We'll also see bamboo corals. We'll see the Hawaiian gold coral, Kula mana mana haumea. It's actually a zoanthid that is parasitic on top of bamboo corals. So that one is a really cool coral to talk about, so I'll definitely talk about it later. We'll see a number of species of plexaurid corals, Muracides, and other yellow plexaurids. We might see some mushroom corals. We'll probably see a number of different types of crabs and bottom fishes. So it should be a really exciting and fun dive today. Lots of really, really cool stuff. And this coral bed isn't very deep. It starts at about 450 to 500 meters. So we're expecting a really short descent time today. We'll be on bottom in maybe 20 minutes.
Oh, wait.
Dan, uphill is to the northwest.
Rob, as the geologist on board, the rock scientist on board. Can't hear.
Okay, Roger, you got that?
Test. Rob, so as the geologist on board, what are you expecting? Because we've heard from our marine biologists. What are you expecting down at the coral bed tonight?
I'm not expecting anything. I think it's just going to be covered in coral and maybe some lavas. Yeah,
It's all
I,
coral.
I don't usually deal with this shallow at depth.
Yeah,
I'm a deep person.
I'm sure. I know
Yeah.
you are. Got to ask you all the philosophical questions once we're doing the deep dives.
Sounds good.
All right, while we're waiting for the front row to continue to get settled, I have some questions for the back row. Since we've already introduced some of us, Data Chris, do you mind talking to us a little bit about how you ended up in the seat you are now?
Sure. So, I, one of my other jobs, I manage. Is that better? Okay. One of my other jobs, I manage a research station on Palmyra Atoll, and we had a volunteer down there who was a navigator here on Nautilus, and she told me about this position opening up, and I applied, and that's pretty much how I ended up here.
What do you normally do on Palmyra Atoll?
So my role is managing the research station, and we facilitate research for institutions from all over the world, mostly to come study corals and seabirds and climate changes.
Do you have any particular type of research that you find interesting in terms of like who the folks who come to the station?
Well, personally, I'm a fish guy, so I really like fish. But the seabird stuff I've been really interested in lately, and watching the coral resilience. We had a big bleaching event this past year, and watching the coral changes with that was really fascinating too.
How did they change?
There was a pretty massive bleaching event, so a lot of them are starting to come back. Some of them are probably not going to recover, but it's been really interesting watching the change, the progression of pre-bleaching, throughout bleaching, and then the recovery phase.
What type of time scale does that normally operate on?
Well, the bleaching started in about October, and by December, most of it was pretty white. Most of it was pretty bleached out, which it wasn't necessarily dead. It was just bleached, and the tissue was still alive, but the symbiotic algae in their tissues was expelled. And then when the temperatures drop down again, then the corals start to get the algae back in their tissues and start to regrow a little bit and recover some of their skeletons that they had died on, I guess. But so I learned a little bit about resheathing, which I had not known much about before, which is pretty fascinating.
What is that?
So when some of the coral tissue dies, the skeleton or the calcium carbonate of the skeleton can be taken over by algae. And then when the coral starts to recover and becomes healthy again, it can reclaim some of that skeleton and start growing on it again.
Some of the skeleton, not just the symbiotic organisms that are with it?
The organisms, the symbionts, are in the tissue
Okay,
of the coral itself, not in the skeleton.
okay. But they can recover some of the skeleton.
Yeah,
as
so
well.
they'll
That's
start
really
to
cool.
reclaim it. Yeah.
I'm sure you're having been at the research station for a while, you get to see this progress in a way that not a lot of people do.
Yeah, I feel really fortunate to have been able to spend a lot of time out there.
Yeah.
And it is some of the nicest corals and nicest coral cover I've seen anywhere.
It sounds beautiful. I would love to visit sometime.
You should come down. It's great.
Science, Chris, you want to share a little bit about how you ended up in your seat right there? Maybe a little bit about ocean census?
Sure. I'm part of the Ocean Census Initiative, and the goal of the Ocean Census is to accelerate the process of describing new species, describing diversity in the oceans. So yeah, that's how everything started. And as a taxonomist, my goal is to identify and classify and mapping their distribution so we can try to understand what we have and how they are distributed in the ocean.
Why is that so important?
Well, if you think about conservation means, and also what how we can better um, use this space, that's essential. Taxonomy is is the base for all the other different kinds of studies that will come from that.
What's your favorite part of the job as a taxonomist and someone who discovers new species?
I think my favorite part is try to solve these puzzles. You know, what we see, and is that a new species? It's not. What differentiates this one from the other? You know, that's the most fantastic part for me.
What sorts of techniques do you use to determine whether or not a certain species has evolved away from another one?
So for sponges, we use everything that we can, but the taxonomy is based on the skeleton of the sponge. So basically,
Sponges have skeletons?
yeah.
What? Okay, talk about that a little bit more. Blows my mind.
So you have sponges with organic and inorganic skeletons. Most of them have a mineral skeleton, and the most common groups you have skeletons made of silica, which is glass.
So we have sponge skeletons that are made of rock and sponge skeletons that are made of glass.
Made of calcium carbonate.
Calcium carbonate, okay.
Yes, or glass, kind of glass, silica.
Okay.
Then you have that really tiny structures make the structure of their whole body. So and you have huge variation of shapes and sizes, and that's what we use to identify them. So we actually you gonna of course get body shape and colors and locations, geographic location, plus the skeleton structures, how they organize in the skeleton, display it. That's what basically we use to identify morphologically a sponge.
Do you need to use any tools like microscopes in order to see how specific they are?
Exactly. We use scopes and compound microscopes. When we are describing new species, you need to also use some other cool tools like scan electron microscopes, so you have really nice pictures of them.
That's so many different kinds of microscopes. What are the different things that they do?
Well, the compound microscope you can see as really small things. It's an optical microscope, and then you can get pictures, images of skeleton organization and all the spicules, and then the electron compound microscope, you use electrons to have better images of really, really tiny
Wow.
structures. And you can see that in a 3D point of view.
Oh, so you get a 3D image out of it too.
It's kind of, yeah, you have a better dimension in terms of what do you see in the optical and what do you see in the scan electron microscope.
Cool.
Yeah.
I would love to see some sponges under the scanning electron microscope sometime. I think things have quieted down a little bit in the front row, so if I could get folks down there to introduce themselves and a little bit what they do, that would be amazing. Let's start with Amber. Amber's toggling some
almost
things.
ready.
There she is.
We're doing some tech stuff over
Oh, I'm
here.
so sorry. That's
No,
all right.
you're totally good.
Annabelle, how about you? Annabelle. I don't think Annabelle has this channel on.
Yeah, it's okay. So I'll start. I'm Amber. I am a lead video engineer, and I've got Marin here with me, who is our intern and learning the system. I am thrilled to be here. I'm excited to see what we're going to see.
We're very excited for you to be here too. Marin?
Yeah, I'm Marin. I'm the video intern for this watch. This is my first dive ever, and I'm super excited. Just looking at Amber and teaching me a whole lot.
Yeah, we're first dive buddies.
Yeah.
Neither of us have ever done this before.
We're in this together.
Marin, how'd you end up in that seat?
I was a really big fan of Nautilus. I used to watch all their live streams during COVID, and just like getting to be a part of the dives was something that I really wanted to do, and I saw that the intern position opened up in January for applications, and I jumped on it as soon as I could.
Amazing. Thank you so much.
I think they're getting Annabelle on the SPO right now.
Who's talking?
Okay. All right, front row is focused because we are approaching the seafloor right now. And as you can all see, the image from the cameras on Hercules have changed a good amount. There's more sediment floating around.
Altitude 48 meters, approaching bottom. That current is going to be problematic, isn't it, Robert? Most definitely going
Was
to be
it a little spicy?
It is nearly inoperably spicy, possibly inoperably spicy.
Yeah, well, that is the challenge about this particular dive site. It is known to have some really strong currents, which is what the corals really like, which is why the bed is so prolific around here.
Yeah, so right now what's happening, just to clue you in, Megan, that the at this point Hercules is fully stretched out, being pulled in the current, and we don't have any control to back up to get settled under Atalanta, and we're still being pushed. Not only are we trying to back up, but we're still actually losing ground. So it is very fast, and I don't know that, we'll see as we get closer to the bottom. We won't be able to have Hercules.
Yeah, it's very flat here. Yeah. There shouldn't be any topography at all. But yeah, it's known to be very quick.
Yeah. Okay, so I got Hercules 25 meters off deck. Argus at altitude, Atalanta altitude is 30, so I don't know if you want to hold there.
Hold. Roger.
And
holding.
Yeah. No, I don't think so either, but at least. And then, so you see how terrain started to kind of show up there in it's flat, you won't see too much, but they're definitely getting some returns in Atalanta. Let's see, the wire cam is what I'm concerned with. How's that in relation to the the transom of the ship?
The wire cam?
Yeah.
it's almost like swinging a little bit.
That's just the swell. It would
Yeah.
be where it is in
It
relation
seems to be straight down. It doesn't seem to have moved very much.
Okay. Do you see what I'm concerned about though? You see how
Yeah.
Atalanta's way over there?
She's
Um,
swung quite a bit, yeah.
Yeah.
You got over now?
Yeah. Roger. Yeah, see if you can back up and descend, but we will try the backup first. Otherwise, this is getting kind of questionable. The Tritech is a scanning sonar that's in the brow of Hercules, so it's only got that 180, whereas the Mezzo will have full around. Yeah. So you can see there, that's Hercules and Atalanta's, that little blip. So that gives you an idea of what you'll see when there is something strong. And that terrain, it'll depend on it. It'll depend on where it is in that slice. So something, it's scanning stuff, but it starts to open up, right? So you're just getting bottom, but you're not, you know, Atalanta's 30 meters off the bottom. Yeah, roger that, Robert. Okay, yeah. So on SPL now, this is the front row. Just for context here, what we're working with is a really strong current, and it's vastly reducing our capabilities with Hercules. So with our two-body system, um, we've got a completely stretched out tether, and um, we've been kind of pulled way off where we would expect to be. So we're lowering Atalanta down at the aft of the ship, but the currents have pulled Atalanta about 75 meters away uh, from that location, and at 500 meters depth, that's pretty significant. So what we're testing right now is if we, while we're close to the bottom, if we have a little bit less current, a little bit more maneuverability, and from there, if we can proceed. Yeah, so we are capable of going backwards right now a little bit. Video, can you iris down on the Hercules HD camera?
Oh,
Yeah, the bed here is all carbonate, so it's pretty reflective.
So right now he's backing up, right? We're just watching the aft cameras and all situational awareness here. It's pretty flat at the moment, but. Roger. So you can see that terrain right there is behind Atalanta, right? Atalanta's facing
look,
it. That's
there's a
expected, right?
Sardinia.
We're expecting the slope to be that direction. And then you can take a look. So Atalanta's altitude right now is 17 meters. So they have some play. I mean, we're not really heaving too much. If we were, you can't have Atalanta too close to the bottom, otherwise it's heaving right towards it. It'll kick up sediment, and at worst it would hit the bottom, right? But we still have some play there. And then you can see in Hercules you're picking up some terrain. But usually we're facing upslope, so we see terrain in the front forward part. But yeah. And now at this point, Atalanta's so low and it's so flat that we're kind of getting we're getting contact everywhere, which makes sense. I mean, from what we're seeing in Hercules, and there's not a lot of like major obstacles, right? But we're low enough now that we're picking up that baseline. And you could adjust the gains so that goes away, but you don't really want to, because we're actually we're pretty low. We've got 15 meters altitude. You want to see that there's some surface there. Trying your luck in autos. Is that the goal, Robert? Trying your luck with the computer? Yeah. Oh, your stick lock back, huh? Do I see only one thruster going? What's going on there? The auto heading was fighting it. That was it, or something like that. Yeah, you see those that blue box over there, and then yeah. Oh, that's weird. That's weird. So what's causing that? Was that the head auto heading? Yeah, yeah. Oh, not that. No, the starboard thruster. Yeah, yeah. Yeah, trying to fight it. Yeah. Yeah. I mean, even right now, you're not really making backwards towards Hercules. You're making backwards, but only. Yeah, for sure, because of the current. Yeah, yeah.
Mm.
Yeah. Oh yeah, there's a little bit of debris there on the starboard side that looks like a trash fish or something. Yeah.
A trash fish?
Yeah. Would be my guess.
Oh, yeah.
Good eye.
We do know there is quite a bit of trash in the ocean, unfortunately. But there's a lot of cool animals, too. So these yellow corals we are seeing are Enelopsamia. They're a sclerotinian coral, so a hard coral. The
yeah,
orange finger
You can see that
is a
Chrysogorgia
Brisingid sea star.
flapping
Yeah.
in the breeze.
Yeah, the Chrysogorgia, that's so fluffy.
Yeah.
And all the black corals.
Yeah, that's really ripping, huh?
The corals seem to like it that way, though.
Makes sense.
I have a question. Is this about the density that we would expect them to be at?
We'll probably see a higher density than this. So right now we're outside of the bed, and that was by design because we don't know kind of where it starts. And then we start to see these high densities of the precious corals. The Enelopsamia is not a precious coral, but as we move along our transect, if we're able to continue with the dive, then we'll be able to see those corals. And my goal with this transect is to be able to document where we start to see those first pink corals and where it gets to be into high density. And at the other end, where do we see that stop?
Got it. Okay. So we're essentially measuring the borders of this bed?
You can start to see Hercules there show up in Atalanta's main HD. See that lights of Hercules? And that tracks here, right? So we're just catching that angle of Herc. So this is what I'm talking about with that DVL deviation, right? You see how those are kind of over time. We would probably, I would expect to see a lot of that drift with the high currents happening. So over time, but for now it's kind of close enough. I can do, okay, Roger, do it.
Turning around.
There he goes.
Ooh.
And then
Is
he's
that
got
a
to
pink
try
coral?
to drive forward.
No, that's a sea star.
Okay.
Yeah, that's a Brisingid sea star. They really like high current as well, and they'll generally always congregate in high areas. So you see we have this one boulder, and you see a bunch of them on top.
Atalanta is.
The pink holes will look a lot like these yellow Enelopsamia, but they'll be pink instead.
So you see how he's getting closer there? So,
There's
now
one of
Atalanta
those
needs to raise up.
Yyyy-mm-dd, purple, that
Otherwise
purple
you'll get a big
one.
loop in the tether, right?
The kind of dusky colored one?
Mm-hmm.
It's pretty.
And they often have symbiotic snake star that lives in its branches. So you'll often see a lot of these corals, especially these Gorgonian corals, have snake stars living in the branches. And the snake star can help clean the coral and protect it, and then the star gets this ability to be up off the bottom where it can capture more food. So they work together in sort of this mutualistic
Oh my gosh, hi
beneficial
Hercules.
relationship.
All right.
See some crinoids.
Yeah, so Robert, the track that we are meant to go before we start to go along it, and Megan also, as we're Yeah, I'll let you know. So the bearing that we intend to go on this, and to be discussed based on current, would be 325. Yeah, something like that. 326.
Yeah, if we think we can do that, that's great. Otherwise, we could pick up and move a bit so that we go through the middle of the bed and just head due north.
Okay, yeah, go along. Well, yeah, that would be like with the current. Then
Yeah.
we I don't know. We'll see what kind of control we have, right? So it seems like laterals are unhappy. Yeah. Okay. So, right. If you do head north, are you able then to have that control fore and aft to be north and south? Yeah. Yeah. But it's at least better than lateral, yeah?
Megan, we have a really good question from online.
Yeah.
Asking about what makes a coral precious, especially if the yellow
Yeah.
coral and the pink coral that we're about to see look really similar.
So what makes it precious is what people want it for. So people want these pink corals for
Well,
jewelry making
from
because their
south to
skeleton
north.
is a very beautiful pink or red color. And that's what makes it precious versus all the other ones. The Enelopsamia, that yellow one, the tissue is yellow, but the skeleton is white. So all these little white bits you see on the seafloor, that's all dead skeleton, because that coral makes this calcium carbonate skeleton that's just like the skeletons of your shallow water corals. So when a coral bleaches, you see the white skeleton. That's what you're seeing here. Their skeleton is actually white. But in the case of the pink coral, the skeleton is actually pink. Other precious corals include black corals. So in the black coral, the skeleton is black. And the gold coral, the skeleton is gold.
It's
So,
gold? How do they get these colors? Like, what makes the pink skeleton pink? And I assume that the gold coral isn't, made out of gold.
It's not made out of gold. It's the gold coral. It's a proteinaceous skeleton that has sort of a golden luster, and you can high polish it. Same with the black coral. It's proteinaceous, and you can polish it. It's almost as hard as a rock.
What does proteinaceous mean?
It's made up of proteins.
Okay.
So it's a hard skeleton, but it's made mainly from protein as opposed to made from carbonates.
And those proteins won't break down up on the surface?
No, it's really hard. It stays very stable. People make jewelry out of it. And it's really beautiful to be able to see these corals. But I think they're more pretty when they're living.
I agree.
Oh, did you? It's a little bit better. We can look at our favorite fish.
Okay,
See that
Megan,
little fish?
So cute.
to catch you up,
Okay.
what we think we can do is work in this orientation where, I don't know if you're looking at either
I've got high pack pulled up.
Okay, I'll zoom in. You can see how much we're being pulled, right?
Yeah.
Up here. So
Yeah, I was worried about that.
Atalanta's currently facing 330, and we're going about 325, 330. That's the direction we would start to pull. Hercules can face into the current in this direction, and then we would lateral towards 325. So
Okay.
we won't be able to face upslope, but we'll be able to lateral.
I think that's doable.
So you can see the heading that Hercules is currently at. We're just gonna do that, but the track bearing will be 325. And that's kind of what we got based on the heavy current.
We got to do
Yeah.
the best we can with
So
what we
your
have.
video will be right to left instead of
Forward.
forward. Yeah.
That might be challenging to get size estimates on the corals because the corals are going to be perpendicular to the current,
Yeah.
but maybe it'll work out. So
Yeah. Yeah, we'll have to see what it looks like, but that's kind of our only maneuverability option. It's kind of
right,
like that or
that or nothing. Yeah.
yeah.
Well, it's
Or
better to have something than
yeah.
nothing, and the video is going to be really important. So
Yeah.
We gotta wait for him. That is good. Let's go ahead and zoom in. I'm on SPL. I am on the training position. Okay, so I'm looking here to make sure that that's all in focus and that my percentage for the aperture is 80. Okay, and then from here, Robert, we are going to go black real quick. So we focused on the tape. We ensured that this was 1B. Now you hit the auto black, and it will do this for a while. It will come back. We're good. Okay, and now we're going to do the white balance, which is you push up here. It thinks about it. I'm going to do it one more time for safe measure. It thinks about it. It's good. Now to save that, we press and hold this. It will beep at us. There it is. Okay, and now I'll come out, and we'll see if that looks like normal colors. And we're looking at this histogram here. This looks pretty up and down. It's a little blue, but I think that's looks white.
I think this should be fine. I'll try. So Megan, if we start right now, we are
Right, so I would say
currently
that's good. Robert, we are good. Thank you.
we're currently about 100 meters, bearing 050 from the waypoint 1. But it would be ideal if we can just start from here and go towards 10.
yeah, that's fine.
Okay.
That should be perfectly wonderful.
Okay, great. Just double checking.
Yeah. That won't really change too much. We'll still hit the middle of the bed.
Yeah, I think so. We're effectively at the same. So I can select this target here, and then that'll give me bearing and stuff. So we got 325.
Yeah, that
So I've
sounds
selected that. So Hercules target bearing 325.
good.
You could also just do that. Okay. So I'll drop a new target here then for our start of, or I'll call it waypoint 1A. How about that?
1A, I like it.
Okay. Pilots, when you're ready, and Megan, when you're ready, we can start this whole crazy trek to 325.
It sounds good.
Okay, yeah, let's go ahead and get moving. I'm going to call that ship move in. We'll start at 0.3 knots because it's pretty flat, and I think we'll maintain that the whole time, yeah? But we'll see
That's
if it
the
feels
ideal.
spicy, then we'll do something different. Bridge, nav. Hello, we would like to track a line bearing 325, speed 0.3 knots. Thank you. So he's confirmed that there, then we just watch over here. There's some gains to mess with. We've also got terrain right in front of us, so that, yeah. Yeah, this is a new visual. Okay, so here he typed in 500 meters step, 325. I saw he had the speed at 0.3. We've got our speed over ground here as we'll slowly ramp up. Go ahead, bridge. The heading's okay for me. We'll see if it's okay with the forward way. If we end up having difficulties, then we can adjust. But right now the heading is good. Yeah, roger that. I don't know, with the wind as strong as it is, I don't know. We only have a little bit of north, but so we'll start from here and we'll see what we can get. Right now it's already, the speed over ground is already approaching 0.2, so I think we're okay for now. Okay, thank you, Paul. So he was talking about, is it okay at this heading? Yeah. 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 Range that down. The ship has just started that movement, so as soon as Atalanta moves, then you can go. Okay, video, it looks like Atalanta has, a camera was changed there on the upper left. I don't know if you can, yeah, we've got the, the A-frame cam. Okay, yeah.
Yeah, that's a weird view.
What dive salvo are we on right now, guys?
What was that?
What dive salvo?
Is this Makapu'u?
So that's regular dive salvo, and then,
Oh, I thought you said
there we go,
dive
we're
steady.
back.
Oh, okay. Okay. So it's what you want to see right now?
Yeah, we're good.
Okay.
Yeah. Yeah, we gotta wait till we get Atalanta to move. Annabelle, if you hold there. So we're waiting for Atalanta to move. The ship has moved, but Atalanta. Yeah. Yeah, there you go. Yeah. Yeah. You might have to tilt your camera down as you go too, but. Okay, so ship's moving. Atalanta will take a little bit to move, and then I'm really curious, honestly, how it starts to move in the current as well, but we'll see over time how that goes. Yeah, that's wise. What's the intent? Is Chris going to be sitting for this upcoming watch, or somebody going to sit with him? Or okay. Yeah, yeah. I'll be here training up Matt, so we'll have that going. All right, so Atalanta's starting to move a bit there at that 325. You can see how Hercules is changing where it is in the camera, right? And you can see Atalanta's got some USBL pings away. Yeah. Roger that. So you can see point 3 is not cooking or anything, but it'll catch up, you know, like we'll see if we need to slow, but I think in the direction we're going, we'll be all right. So Robert, are you currently doing this computer, or is that just drift? That's auto XY, Roger, yeah. So because we're lateraling here, we're going to be eyeing up Herc's starboard cam just to see if there's any obstacle, but you know, as we go in that direction, we'll also look in Herc's mezzo, which is upslope, you know, because that's the way we're going, and we'll see if there's going to be any boulder or anything in the way, but it's probably going to be a lot of this. So easy to navigate. And he's got it in auto altitude there, so it's not like we'll hit the seafloor going upslope, it'll raise up with us. And Megan, as we just get started here, you're good with that camera angle and view and lasers and everything?
Yeah, so we got the lasers on. I like this view. So if we could just kind of keep it like this, I think we're good.
We'll do, yeah. Robert, if Atalanta keeps trending in that direction, so we'll have to go a little more. Yeah, yeah. Just so we don't end. There we go. So you change your heading to the starboard just a few degrees. There we go. Yeah. Well, you can see, so you see the ship's track, that line right there, and you see Atalanta. They're both about the same, right? So if you're looking on that nav screen, Annabelle, over on the left, yeah, you see that track is about what we've been doing, so that's about right. And then Robert will adjust where he is based on that. Yeah, how's that speed? Is that okay? Good. So you can see the ship, because we actually have some surface current going this way, we're still maintaining against that wind, but rather than fight against that current, it's just kind of going straight, and we're not like going in this direction really, we're just kind of like letting it drift. So it's actually doesn't have to work that hard. He was asking about if we need to change heading, and if we were to change heading into the north, we'd have the wind more abeam, which would help us go, but it might help us go too much. Yeah, so. And then you can auto center this on like Argus if you want, but I usually just tend to move it myself because I like to keep all these vehicles in view, and sometimes that prevents that. But one thing I did when I popped in here was this tab right here, the length that's the trail length for this. So all of this right here is, you can change it to be the USBL or whatever, but right now it's on the dead rec.
So let me know when we're like
that's
ready
like a
to
long.
go.
We're going.
Oh, we're going.
Yeah, we're
Did
moving.
someone log a start a transect?
That would be up to you guys.
Okay, I will log that.
Yeah.
Okay.
Yeah, this is the speed. It'll be this fast is about 0.3, and it'll be in this direction. So Megan, I'd say we probably traveled about 30 meters so far.
Okay.
If that helps you, or the start of transect will be a little prior.
It's close enough.
Yeah. So this yellow, is that zoanthids colonizing these?
No, those are actually Enelopsamia, so they're hard corals.
Okay.
So they make a calcium carbonate skeleton, and their tissue is that bright yellow color.
So they have a kind of same tissue color as some of the zoanthids we see, yeah?
Yeah. I think there's a nice little hymenaster, a slime star, that pink. No zooming.
No zoom. You got as auto as you can get over there, man. Yeah. So once you get something like this dialed in and it's kind of observing, you don't want anyone to get complacent, because then suddenly if you stretch the tether out or something, things, especially in a high current environment, this would go south. But if we keep it all as is, we'll be good. Yeah.
Yeah. It is strong here.
Yeah. Yeah, it took a second to get situated. Until we figured that out, it was going to be pretty questionable. I mean, even still, like you said, we wanted to go forward rather than sideways, but this is what we can do.
It'll just be a different way of thinking about it, you know?
Yeah.
Everything will be scrolling past like a landscape instead of driving over it.
You'll just take your video and you'll rotate it 90 degrees.
Yeah, okay. That'll be fun.
Yeah.
Just
Yeah.
Yeah.
Widescreen, right? Yeah. More ID time. But we're lucky here because the slope is it's not too steep though. We're not going to bump into anything. We'll keep an eye out for the stray boulder here and there, but
stray boulder? Oh.
Every once in a while, be something.
Yeah. There's always some rocks, Can't control that. But generally it's going to be this flat hard pan.
So at this pace, we'll make it to 0.10 around 7 hours from now.
That's about as much as what we have, right?
Yeah, I think we're going to be on deck at 1600, unless that changes. I don't know how much wiggle room there is.
I don't know if we need to make it all the way to 10.
Yeah.
10 was just my ambitious goal.
Yeah, so we'll get...
as long as we get across. Yeah, we could probably move a little faster. I just want to do what's comfortable for everyone.
Yeah. It's up to you guys. I could bump it to 0.4.
Like now, since everything's scrolling by, it seems so much slower than when we're flying over it.
Right, yeah. We'll see. We'll keep this going for a few, and then we'll...
Yeah, this beginning part here, since we're not seeing any precious corals, is just sort of, it's not really that important. I mean, it's important to note where they start, but right now we're not seeing our target organism.
Oh, so that's not these yellow ones that we've been seeing?
No,
yet?
no,
Okay.
it's not these yellow ones.
They're the pink precious corals, yeah?
Yeah,
Yeah.
we're a little bit deep, and that was by design. I wanted to start
Sure.
outside the bed and then move forward.
What is the depth band for this organism?
Usually about 450,
Okay.
but they have been known to go to about 500, but it gets really sparse.
Do they go, what's their shallowest?
Like 350.
Okay, so pretty tight 100-meter band there. Is that common for corals to have that kind of tight of a depth?
That's what they're like around here, but you remember from other dives that we see pink coral at really, really deep depths as well, but they're different species. So here we have two main species. We have Hemicorallium lawense and Pleurocorallium secundum. And they're really hard to tell apart. They look exactly the same on video, so. You'll probably also notice that there's a lot of fish around the bottom, so the fish really like this type of area here. Oh, there's a sponge for you.
It's a sponge.
It is. It
Chris,
seems
what
like
sponge is it?
a glass sponge, one of the stalked Bolosomines.
And the fish are like this type of habitat, so we see quite a few of them, especially where there's like little overhangs where they can find shelter, and they do a lot of hunting on this flat hard pan. So we've been seeing some rat-tail fish, like the one in the background there, and some green eyes have been sitting on the bottom. We also might see some scorpion fish, some ectophids, which are lanternfish. And when we first got on bottom, we saw Slidenia reminger. Those are goosefish, related to the monkfish. They're a type of angler, so they got a lure, and they have that little grumpy face. That's just so cute. You gotta love it.
It's like some of our crewmates.
Just, little grumpy faces.
But so cute.
But so
and
cute.
We gotta love it. Aloha everyone, this is Daniel Kinzer, communications lead on NA-163, and just stepping in and taking the SCF seat for a few minutes. Give our awesome team a little bit of a break. But nice to see we've gotten settled down here on the seafloor in these ripping currents and still getting some exploration done. That's awesome. Good work.
It was always a unique challenge to dive here. Dive anywhere, actually. Lots of pressure. It's dark. We got to be prepared for a lot of different types of situations.
Absolutely. It's one of my favorite things about being on board the Nautilus is watching the team in action, dealing with all those different pressures, not just the pressure of the deep sea, but all the different pressures of making this work. And you all were quite impressive to watch. I was sitting back in the studio and had a good view. Well done. Megan, to the extent you had hopes or expectations about what we might see on this dive, how has this been matching up to those?
So we were actually outside of the precious coral bed.
We are, yeah.
So we're not seeing those pink corals that we were expecting, but that's because we're a little bit deep. And as we head up slope and across the known bed, we'll see more and different varieties of corals.
Okay.
But right now, we are seeing what I expected. We've got some Chrysogorgia, some Enolopsamia, a number of different types of fish species hanging out in this area.
Yep.
And as we get a little bit shallower, we'll start to see those pink corals, the bamboo corals, more plexaurids coming into play.
Yeah, awesome. It's always a pleasure listening to our ROV pilots. So talented, amazing at the work that they do. And
oh, come on, you're inflating my ego here.
So eager to throw each other to the wolves. It's really great. It's a lot of fun.
You know the hardest thing for an ROV pilot to do?
Tell me.
Watch another ROV pilot operate.
Yeah. I know what else is hard for Dan. Watching me navigate.
Megan can navigate great. She just gets sidetracked by the beautiful biology.
It is beautiful, and I like looking at it.
Speaking of that, where's the ROVs? We flew right off the map.
Right under the ship.
Find your ROVs, Dan. Come on.
Yeah. Well, that's the navigator's job.
Oh. Well, also Dan. We've got a lot of Daniels in
Lots
the...
of Dans. Oh,
got
yeah.
a lot of Dans in the control van today.
And Dancing Chris's.
Yeah.
Dan van?
Yeah. It's the, oh, this could be the Dan van. I like it. This is good.
It's funny how that works out when you get like multiples of names.
Mm-hmm.
And they all have very different jobs. So you don't know who you're going to get.
Yeah, you got to say like Navigator Dan. How's first time navigating in these strong currents off Makapu'u here on Nautilus? You having a good time?
You're not on SPL, man. You're
ROV Dan, can you help Dan find his switch?
You're muted there, matey.
Yeah, first dive is always hard to get
the
reoriented.
Mute is the one right to the right of SPL.
Push it
Let's
up.
see where it says it. Yeah, push that one up.
Teamwork, making the dream work in the
now
van.
Now you're live.
Having a great time navigating. There was a steep learning curve with the launch and dealing with the currents, so it's nice to have a hand with that from Rini. But I'm starting to get the hang of understanding what I'm looking at here.
You're doing a great job. Yeah, it was great watching you and Rennie and the ROV pilots kind of figured that all out. It wasn't an easy first dive, so you did awesome.
Dan, what kind of delta do you want as you start coming underneath a little bit?
I see you got an extra screen
Robert
down
was
there.
having me hold around 17,
bonus.
Yeah, you can come up a couple meters.
Roger.
I'm all about more screens. Just add more screens. People ask how many screens there are in the van. I don't know.
Not enough?
It's a lot. It's too many.
It's
not enough.
Too much screen time.
All the screens are important. Got a little overhang here. You'll see the fishes like to live underneath that overhang. They really like this little topography. Oh, and there's some heterocarpus shrimps. Those are particularly tasty. It's lunchtime.
It is almost lunchtime. Actually, it is lunchtime on board. Next watch is down there getting their fill. They'll be in here in just about 15
Look
minutes.
down just a little bit with your Atalanta camera.
Roger.
Rob, has there been anything of interest from a geologist point of view? Anything you've noticed on this dive that you haven't gotten to share yet?
Just carbonate.
Yeah, just carbonate. Lots of carbonate.
Yeah.
Yep.
There's some Poecilastra for you. The plate corals, or the sponges, plate sponges. Lots of Bracyngids. I like them, they're really fun. They're filter feeding sea stars, so they like put their arms up in the water column, and when they're like that, that's their feeding posture. And you'll see them collect little bits because their mouths are on the underside of the star.
Yep.
Do you see many basket stars around here?
Yeah, we do see basket stars. They tend to be on
I heard
top of
little guy and I
corals.
was like, you must be in here.
So we might see some as we get closer to the higher density coral areas.
They scare me.
Is that a sea urchin?
Yes, that is a sea urchin. It's a Histosideris. Yeah, these are all very typical animals for this area. That little fluffy one is a Chrysogorgia. You'll notice some black fuzzy looking ones. Those are black corals. And the fish on the bottom right in the middle there is a green eye, Chlorophthalmus. So when I get this video in the lab, we'll be able to use my annotation software and log all these animals.
Oh,
We now
awesome.
have the capability to make localizations on the video, so we can feed the imagery into FathomNet, which will help and train models to learn more about annotating video, so that our AI models can identify these animals or help us identify these animals in video. It's going to be a long time coming before they can do
3
my
meters,
job,
please.
but as we feed it more
Can
and
we
more
step the
data,
boat,
it's getting better at figuring out what is an
Change
animal
the
and what
boat's
isn't an
course
animal.
over ground to the west a little bit, just to get Atalanta to the west of Hercules there.
Yep. Like 5-degree shift.
Whatever it takes, we should have
Get
some.
it west of Atalanta.
Yeah. So these are 10-meter boxes here.
Okay.
So Atalanta should be about right here right now.
Okay.
I can. I don't want to change my video transect, so you're gonna have to.
There's another Slidenia.
The course
little
over ground's
goose fishes
currently
on the bottom.
325. I would make it 315. Another thing you can do is
And then on that Enolapsomnia in the middle, that brown, that's a crinoid. Or it could be a basket star. Actually, it is basket. There's your basket star.
There you go.
There it is. Called it.
Kirk's course over ground.
Deep sea prophet over here.
Yeah, 315. So you're gonna have to change it to like 310, something like that.
I just want more of a warning
And
when the
some
basket
tiny
stars are.
sponge balls.
Let's just try 300.
Oh yeah, the way they move is like
300.
really fascinating. And a little creepy.
Very creepy.
You want to go on the comms now?
Bridge nav. Can we change our course over ground to 300?
Roger.
Roger that.
Rob, some of our viewers on the internet are wondering if you could give them a wave because they can't see you. They don't know where you are. I don't know. And
Well,
So Megan, I'm gonna
put Bobby in the
hold
corner
position here for a minute.
Okay.
I believe the boat's
See me
holding
now?
position.
I think that's good. I think they'll see you.
Are you all right with that? That's
Yeah,
not gonna
that's
wreck your
no,
transect?
it won't wreck my transect.
You want to stop away?
I'm
I
gonna
can't hear you, mate.
estimate area
We're in a hold
surveyed,
position.
The boat's
not using
changing.
time.
We're changing course. We're not stopping our way.
He said he was gonna hold position for a minute and change.
Oh, okay.
I'm watching his speed there. It's right, currently 0.1.
Nav, go ahead.
Yeah, there's a
Uh,
couple different ways you
is-
can analyze a video transect.
He's all stopped. Just
People have
Just
split
have
up
him do
transects
a,
based
just
on
have him do a
time
20
and
meter
video.
move west.
So like they take 10-minute chunks and they analyze it, and that's how they
Bridge
separate
nav,
it.
can you move 20 meters
But as
to the
we
west?
get better navigation data, we can split it using our nav data.
Yeah,
So
I
as
love
long
it.
as we're staying
Copy,
and
thank
moving
you.
in a straight line, it makes it easier. That's why we're not wagging back and forth or moving up and down. We just kind of want to keep going in the same direction with the same video settings.
Yeah.
That way we're not over like annotating one area that we've looked in high depth and then just really sped by another area.
Sure. Balanced inputs into the AI. We don't want to make it too hard on the AI. It's only artificial after all.
Well, we want to be able to do statistics, and so we want to keep things as you know, rote as possible,
Yeah.
so that every section of the transect is equal in the way it was collected.
That's right. One of our viewers is mentioning Fathomverse, a new game I think Embari and others have helped to put out about getting citizen scientists to help identify and train models to help.
Exactly, and so if you guys are interested in that, definitely download the app. It's free. It works on both Android and iPhone. And I even have it on my phone. It's pretty fun. It's very soothing. It's got some really nice music
Bridge nav,
background.
copy that, thank you.
And it does train you how to identify these animals, like little blurbs about what are the key features of an animal in order to identify it. And then once you've done the little training module of looking through the information, you go on a dive just like this. And you're a little ROV swimming around and an animal will pop up and you click on it. And then you have to decide if it's one of your target species. So like today, we're looking for the precious corals, the pink corals. So that's what we're looking for. We're not seeing it here, so we wouldn't put that in our basket. But when we do, we'd be like, yes, this is identified positively as this animal.
It's a fun game.
Yeah, it's really fun. And all of that information does go into helping train our AI and identify all this data. We have so much video data from the seafloor and not enough people who have expertise like myself to do the identifications. So being able to use citizen science to help identify where is an animal, where isn't an animal, put it into major groups, we can get more of this data processed. And then people who really enjoy this work might want to join a lab like my lab, the Deep Sea Animal Research Center, and learn more about this and do actual video annotations and come aboard ships like the Nautilus and collect this data.
I love it. All the pathways to ocean exploration and ocean love.
Can you?
And like even if you live in a landlocked area and you can't go to the ocean comes to you through your mobile device.
That's right.
It's really fun. Like even someone who does this on the daily as a job still plays the game. So
I like that. That's pretty funny. We do have a watch change coming up, so you might hear a little bit of a changeover, and a new set of navigators, pilots, video engineers, science team. Megan, you going to stay in?
I'll probably go get lunch, but
Get
yes,
some
I'll come
lunch.
back.
That's a good idea. And yeah, we've got a whole new watch. The 12 to 4 watch is making their way in. The 8 to 12 will go and enjoy some lunch. And you'll probably hear from Megan again after she's done. Probably not from me, but it was nice being back on SPL for a little bit, and we appreciate everyone for tuning in, and hope you'll stay with us as we continue along this transect and enjoy this exploration off Makapu'u. All right, aloha everyone.
Just doing an audio test. Okay, we got you.
Aloha nui kako. E nā hoa makamaka o ka aina. Ke la pae, ke ia pae. Nā mamu halo. E ano mako. Ma ka moko nales ke ia manao. E mea luu kaini ma waho o makapu. Mokupuni o waho. E kilo nei ka papuku o ka moana. O ia no ka mako ke ia manao. Ke imi nei i nā ano mea liki ole e ulu nei ma nei. Aloha everybody. This is Mahina Ho and we're currently outside our Makapu on the island of Oahu. Searching for the Makapu precious coral beds. We've been down for a few hours now and Aloha kako. Hui. Oops. All tangled.
Okay, this is Nav. I'm on comms on SPL here, talking to pilots. There we go. Oh, I've got you there, Chris.
I gotcha.
You don't have me, so that bottom one, yeah, you want to crank that up. Yeah, so that bottom knob that you were messing on the right, the far right, yeah, that's your main volume. There you go, crank that up. Yeah, because they have a different headset that's like a different dB or something. Okay. All right, we're back in it. So yeah, Chris, although we're tracking a line at 325, you'll notice that Atalanta is seeming to be drifting more and more westward. I think that there's a reduction of current at this depth. So that's why you'll have to keep doing those jogs forward to kind of keep that distance there. But yeah. So it's always good to check, you know, with your DVL, USBL camera that it is all making sense, you know. We don't change anything typically, but sometimes, you know, oh, somebody cycled this thing and the offset is wrong or something like that. So, yeah. Yeah.